All right, yeah, so I was on a plane 8 hours ago working on a slide deck for you guys. But I think it's really complete. It's a little bit over rather than under, so it'll take us probably the entire 2 hours to get through this. Yeah. Yes, Tuesday, first class day. All right, I'll start off with a question. Let's see how you guys do on this one, okay? A little practice question.
Yeah, start you low.
Size 26 font for those in the back.
Is that Calibri?
It's Arial, sir, it's Arial.
Throw it out. In clusters, yeah, big.
So a lot of people are saying the right letter, which is cool. Some people look really like confident. Some of you have inscrutable expressions that I can't interpret. I'm more worried about you guys. Okay. For the latter, what are your thoughts? Okay. This is an example of a second order question. It relies on you knowing what the organism is. So what is it? Let's start with that. What is the organism today? Staphylococcus aureus. Okay, so if we got that nice little layup, then the second part is pretty easy, right? Because we know the morphology, gram-stain morphology of Staph aureus, right? So gram-positive peroxide clusters, V, pretty easy. Okay. So just because it's a second order question doesn't mean it's hard necessarily. All right, so let's kind of get into some slides that perhaps you should focus on. Just like the one that I just set up for you guys. This is important for a couple reasons. A, it reinforces what that gram-stain morphology is, but also it kind of walks you through the initial algorithm for identifying staph, distinguishing it from strep, the other gram-positive. So what are some ways that we distinguish staph from strep? Catalase. Yeah, so which is catalase positive? Staph, and then, yeah, catalase negative would be your streps. And then what about, less specifically, if you were just to look at the morphology? Not capsule, just like clusters versus chains. All right, and strep is which of those two? Chains, with the exception of one, strep pneumo, which just kind of tends to be a little bit more, yeah, in pairs, lancet-shaped and in pairs. It can be in chains, but it's the one that usually is in pairs. Okay, so clusters, oftentimes for staph. What about if you have the need to distinguish the most virulent member of that genus, Staphylococcus, from less virulent members? There's another biochemical test. Coagulase test. Yes, we talked about coagulase as a virulence factor, but we also use it diagnostically. So if it's coag positive, what would that tell you? Staph aureus, right. Okay, so then we have a bunch of other, like 12 or 13 clinically relevant coag neg species, but we only really focused on 2 of those. What were the ones that we talked about? Saprophyticus and epidermidis, yes. Okay, good, pretty good. So far, so good. All right, so we talked about some skin and soft tissue infections associated with Staphylococcus, particularly Staph aureus. Some of these invasive enzymes, this should kind of be old hat. You know, we have the same major players, know what they do. When we're talking about folliculitis or things like STI, or hair follicles or sebaceous units, those sebaceous units that get colonized and then inflamed, that can be an entry point for deeper infections. So some of these invasive enzymes are important for this. This cartoon is showing some of those bacteria starting to cause inflammation around a hair follicle that can eventually, grow into an abscess, which is a localized bacterial infection. So don't take this stuff for granted, just be comfortable with it. There's coagulase. Again, what does coagulase do as a virulence factor for Staph aureus? Yeah, it forms more or less a protective clot or barrier around the bacteria to prevent it from being cleared from the immune system. Yeah. Okay, familiarity with these infections caused by Staph aureus, the infections themselves, which are broad ranging, everything from pneumonia to pericarditis, sepsis, bone infections, all these skin and soft tissue infections that we talked about. But also the things that aren't bona fide infections, where most of the damage is caused by the toxins produced by Staph aureus. So most of these, see if you have a pretty, how comfortable we are. What's the primary toxin associated with Staphylococcus toxic shock syndrome? TSST1, toxic shock syndrome, toxic 1. Good job. What about scalded skin syndrome? What's the primary toxin associated with that? Exfoliating, yeah, exactly. Which are exfoliated toxins. And then the food poisoning form that causes gastroenteritis.
Which one? There's three. Well, there's 4 possible. What's the worst one?
Fantastic, okay, all right. What's the most common one? Okay, and what's the one that can be weaponized?
B.
Love it, okay, so type A is the most common one in gastroenteritis, and type B is the one that can be weaponized. You need to just kind of Staph enterotoxin B is the one, SEB. That's the one that's associated with biological weapons. Very good. And then there's also C, D, and E, and G, and no F. Okay, very good. Cool. Skin infections, we talked about those, just, kind of be familiar with them. Know the difference between a furuncle and a carbuncle. Carbuncle is just a deeper infection. Furuncle, impetigo. What other thing causes impetigo? Strep pyogenes, yeah. So not really distinctive, are they? You have to be able to rely on lab support in order to distinguish the etiology of impetigo. Toxic shock syndrome. Know that TSST, that toxin that mediates this disease, is a superantigen. So these systemic effects that you get with toxic shock syndrome, like the high fever, vomiting, diarrhea, hypotension, the things that can lead to death, lead to fatal outcomes, that's associated with a superantigen that this bacteria can produce. Okay. Scalded skin syndrome, we talked about a little bit that exfoliating toxin is working on this intracellular connective protein called desmoglein, and it's cleaving that, and that's causing disruption of the outer layers of the skin. In addition to the lab, you have that positive Nicole C sign you want to watch out for. Which demographic is usually most commonly affected by this toxin? Young children, yeah. Good. Okay. And then the foodborne one that we talked about as well, just make sure you glance over that. Or study it hard, rather. We talked about a little bit of antibiotic resistance mechanisms for Staph aureus. We talked about VISA, or VISA, and then we talked about VRSA a little bit. What was VISA? Tell me a little bit about VISA. What are VISA strains? What's the primary mechanism from which they can have any resistance to anything? Yeah, it's a thicker cell wall, so it takes more drug to get that effect. When you have VRSA and you have high level resistance, you need a mechanism that makes it refractile, et cetera. So with VRSA, the mechanism here that was covered in this slide and this slide, which I know we're busy slides, but kind of summarize what's going on, and can somebody tell me what these VRSA strains can do? Yeah, so the pentapeptide that cross-links the magnam peptidoglycan layers in the cell wall. The last one is an alanine, so it's a diala, the 4th and 5th peptides. These VRSA strains have an operon, it's called Van-A operon, that they get from another bacterial genus called Enterococcus that causes them to produce diala-D-lactate and then make a messenger longer binding. And so kind of look over, make sure you conceptually understand what's going on there with that operon that can be horizontally transferred from different bacterial genera. And then those coagulating stuff that we talked about. Not a lot of virulence factors. Opportunistic infections, it's important to know. These tend to be the most common ones because they're on our bodies. I said focus on epidermidis. Where does epidermidis usually colonize human hosts? Skin, yeah, where else?
Posterior.
Yeah, anterior nares as well, yep. And then Saprophyticus is more likely to colonize what part of the body? Yeah, yep, vaginal tract. And what kind of implications does that have when you start thinking about diseases caused that Staph saprophyticus can cause, knowing that it colonizes UTIs? Okay, very good. So when we're talking about UTIs from bacterial causes, remember when we were in deck, so we're still in deck 1, but we're in deck 3, we were talking about all of those gut bacteria like Providencia, Klebsiella, and Proteus, and all those things. A lot of them can cause opportunistic UTIs when you have contamination of the genital urinary tract from those fecal flora, right? But the big two that you think of when you have a patient with a UTI, what are the two big major bacteria that you think of? E. coli, and then Staph saprophyticus. Yeah, but many other bacteria can cause it. So about 75% of UTIs usually implicated with E. coli, right? If you have a person who has a UTI and, I don't know, this is a little bit of a contrived scenario, but the lab tells you that it's gram-positive cocci, are you going to suspect E. coli? No, because that would be what kind of morphology? Gram-negative rods. And it's contrived because you probably see some E. coli in there regardless, but that's something you have to quantify. Okay, and then these complications, make sure you have a look over some of those complications with Saprophyticus. Epidermidis, so opportunistic infection, but know that it does have that ability to form biofilms. So because of that, you can have conditions like endocarditis, biofilms that form over prosthetic joints, things like that. IV catheters implicated in that. Those can cause some serious infections. All right, that's the staph deck. So gram-positive cocci, deck 1 staph. Moving on to deck 2. Similarly, just to kind of get us going, classification for strep is a little bit complicated, so I want to make sure we understand how this works. We talked about two major ways, old ways of categorizing streptococci. What are those two ways broadly? Hemolysis pattern and Lancefield grouping. Okay, cool. With the Lancefield grouping, what's the principle of Lancefield grouping? C substance, and where is that located? In the cell wall, exactly. Okay, good. All right, so take a look over this. The organisms we hit hard, the super virulent species of Streptococci would be things like Streptococcus pyogenes and Streptococcus agalactiae. What is the Lancefield grouping for Streptococcus pyogenes? Group A, yes, group A strep. And the Lancefield grouping for Streptococcus agalactiae is what? Group E. Okay, very good. And the hemolysis pattern for both of those is what? Beta hemolysis. Excellent. Okay. All right, got a question for you guys. I did hear some wrong answers, but the right answer is what? E. Okay, good. Yeah, so we just kind of talked about that, but it's beta hemolysis if we're talking about Streptococcus pyogenes. So if you're a little bit rusty on that or just want to hit it hard, just make sure you kind of look over this and this slide a little bit. Review those slides. So we see a couple things that we've talked about before with Staph aureus, with a different organism and a different genus. Strep pyogenes causing some of the same skin and soft tissue infections. Impetigo, erysipelas. We don't so much associate furuncles and carbuncles with Streptococcus though. That's more of a staph thing, so that's notable. Some really deep tissue infections that are caused by strep due to those plethora of invasive enzymes that it produces. It produces exotoxins capable doing a lot of tissue damage. So you have necrotizing fasciitis and you have myositis. Infections of the fascia and the muscle tissue. This is a good slide to study just to kind of get a sense of what the symptoms and what the presentation might be for necrotizing fasciitis. Group B strep. Be familiar with this. The fact that this can colonize women asymptomatically and cause what kind of problems? What are we more concerned about? Group B strep. Babies, yeah. So babies would probably pick it up if they are born to a mother who's colonized. And what types of problems might this cause in a newborn? Meningitis, yeah, meningitis and sepsis, pneumonia, yeah, exactly. This is a really hard question, but why not? Can you guys name the 6 bacteria that we've talked about so far that cause meningitis appreciably? I mean, lots of things can cause meningitis, but the major things.
E. coli.
I was like, no. Yeah, so Listeria, E. coli, neonatal E. coli. Which one's John? Neisseria meningitidis.
Hip?
Yeah, what's hip?
Haemophilus influenzae.
Wow, Siri just answered that. Yeah, so Haemophilus influenzae, neonatal meningitis, E. coli, Listeria monocytogenes, strep pneumoniae, we've got group B strep. And yeah, Neisseria meningitidis. I might have, was that 6? We just read off? That was 5, okay, well, we'll hit the 6th one, whatever that is, but make sure you're familiar with those agents of meningitis. So yeah, with your B strep, in terms of preventing newborns from acquiring this infection, what's our primary method of mitigating it?
Penicillin at the right time.
Yeah. Screening so we can treat with antibiotics if we identify somebody who's colonized. Do we have a vaccine for GBS?
Yes.
We do not. We do not have a vaccine for GBS. So screening is our primary way of mitigating this infection. All right, strep pneumo, another one of our causes of meningitis. Important that this is one of the ones that can cause meningitis in the adult population that we associate with adults, and make sure you remember that. But this whole triangle is really good. Very common infections, everything from runny noses, which causes a lot of ear infections, mastoiditis, which is the bone that can be invaded if you get a really severe otitis media case, sinusitis, eye infections, pneumonia, bacteria. Just be familiar with all these things that strep pneumo can cause. Okay, so moving on to the 3rd deck, moving along. All the way up to E. coli. There was a slide that I think, I didn't put it in this review deck, but you should probably study it. I've tried to hit it a couple times already, just see how we do. So I keep drilling you guys on the pathotypes of diarrheogenic E. coli. And we broke that up, so there are 5 pathotypes that hopefully you can just kind of rattle off. The 3 non-invasive pathotypes are what? Super. Okay, I think we got that. Okay, EPEC, EAC, and ETEC, very good. And then the 2 invasive pathotypes. Outstanding. All right, EIEC and STEC/EPEC. With EPEC, what are some things that we associate with EPEC, just as mnemonic devices? Yep. Yeah. In terms of the pathology, no toxins produced, really. So we're talking about a pathology where you have that bring your own receptor, it brings its own receptor and causes this damage to the enterocytes, and that causes diarrhea, destruction of microvilli. So watery diarrhea in infants, pedestal formation, cytoskeleton, rearrangements of those target cells, things like that. I'm just kind of spottily going through these, but you should absolutely study all 5 of those pathotypes and be familiar with them. For enterotoxigenic E. coli, we have another watery diarrhea, another non-bloody diarrhea. You might think it's difficult, and it is difficult to distinguish from like EAEC or EPEC, but how might you be able to distinguish that in the lab if you wanted to confirm that somebody had ETEC? Toxins, yeah, but like how in the lab? You can do PCR where you look for the genes themselves, the LT and the ST, or what else potentially, if you just want to know if it has those toxins. Maybe like an immunoassay? Yeah. Okay. And then in terms of the types of populations that are most commonly affected that we see, what kinds of groups and populations are affected by ETEC? Yeah, travelers, exactly. Alright, the STEP. This, we do get outbreaks here in the United States. It's a good one to be familiar with. It is super pathogenic. It can be. It can cause some really severe diarrhea. Big outbreaks associated with contaminated beef, restaurant outbreaks, food chain outbreaks, unpasteurized milk, fresh produce, all that. In terms of diagnosing definitively, you could look for what? The Shiga toxins, yeah, STX1 and STX2. And then in terms of the presentation, is this invasive or non-invasive? Invasive, okay. So those toxins are doing a lot of damage, killing the cells, disrupting the intestinal barrier, and as a result, you get what kind of diarrhea? Yeah, you get bloody diarrhea with this. So be familiar with those 2 slides. I split it up into 2 slides. And also this particularly virulent strain that we hear about a lot that's implicated in foodborne outbreaks is E. coli 0157 H7, where earlier in this deck we talked about some of those antigens and what they mean. What does the O refer to? 0157. Somatic, yeah, the O antigen, and that's used for typing purposes. So 157 is the O antigen 157, and then H corresponds to what antigen? Flagella. Okay, cool. And then what was the third one that's not? Okay, and that is what? Capsule. Okay, very good. A really important sequela or complication that can happen with infections as a result of STEM and EHEC is what? Pus. And it is what? What does that stand for? Hemolytic uremic syndrome. Very good. Moving on to salmonella. So salmonella causes, I don't want to say normal gastroenteritis, but it is a common cause of classic normal non-typhoidal salmonellosis. But you can also get this very severe presentation called typhoid fever. What are some of the signs and symptoms of typhoid fever? Rose spots. The fever is really, I know it's not helpful if somebody has a fever, but that's distinctive for typhoid fever. Let's see. And those rose spots, would they be apparent right away? No, the bacteria would have had time to disseminate, replicate, and that would be causing those spots on the trunk if they're present at all. All right. What do you guys think? Give me a second to read through this. would agree, yes. Alright, so what are some of the things in this question that were making you think it's probably salmonella typhi as opposed to some of these other options?
Travel.
Travel. Okay, so let's talk about travel real quick. Can that be helpful for your diagnosis? Do we get a lot of typhoid fever in the United States? No. Nah, not really, no. Asia, Africa, some hot spots for that. In terms of the symptomology and presentation, it sounds like normal, well, not even diarrhea, abdominal pain and constipation, but the rose-colored rash is not normal for other types of gastroenteritis, so you should be thinking something like typhoid fever. Enteritis, that is associated with what? Normal gastroenteritis, NTS, right, non-typhoidal cellulosis. So you would have symptoms probably like abdominal cramping, you know, just normal gastroenteritis symptoms like that. A 157H7, just talking through how we eliminate some of this stuff. Not consistent with the rash. Most of these you can kind of rule out because of that rash. Dysentery, Shigella dysentery. What kind of presentation would you associate with Shigella dysentery? Okay, and where might you encounter dysentery?
West Africa. West Africa. Like, it's not really Asia.
Yeah, I mean, outside the United States for this, yeah. And we do have that slide coming up in a second there too. What about Vibrio parahemolyticus? What do you guys know about that?
Well, near the water.
It's what?
It didn't need any raw shellfish.
Okay,
It wasn't near the water.
okay, but it's still like, could be, you're ingesting contaminated water, so that's possible. But what kind of symptoms do you associate with Vibrio parahemolyticus? Gastroenteritis, yeah. And can you get that in the United States? Or in the office? Yes, absolutely, absolutely. What's regions of the United States do we associate?
Our region.
Our region, yeah, so the Gulf Coast states primarily, yep. The East Coast. Okay, talked a little about, so we're still kind of talking about typhoid fever, but we're now talking about a carrier state where the patient does not clear the infection. And these bacteria will tend to go into a couple places, hide out, be sequestered, and stay for long, long periods of time. Where would be some places that those bacteria would hide? Gallbladder and biliary tract, yep, very good. Alright, here's that slide of, yeah, so somebody said West Africa, Central America for dysentery. Most severe of these four is what?
Dysentery.
Tends to be the most severe species. Least severe tends to be Sonane. Yep. And the one that we more commonly encounter in the United States? Sonane. Okay. So it doesn't mean we can't get patients with dysentery, but oftentimes they have travel history. So that's always important.
On the test, would you have the name and the group or just?
Yeah, I'll just have the name probably. Yeah. In terms of transmission, so primarily person to person, but given the fact that you have a really low LD50, there are implications for that. Yeah, so you can spread kind of in close proximity to other people who have this infection because they're shedding bacteria in feces, and that can be aerosolized. So with some of these really bad cases of shigellosis, regardless of the species, you can have inflammatory diarrhea. So you can find things like neutrophils and other immune cells in the stool, as well as red blood cells, erythrocytes in the stool, and pus. So that would be helpful for diagnosis. Cholera. What parts very infrequently, usually associated with hurricanes. What about transmission for Vibrio cholera? What is the primary transmission? Ingestion of contaminated water, exactly. Is person-to-person transmission possible? It is, but it gets more efficient when you have passage through people and it gets adapted to human host. This is an environmental bacterium. In terms of mechanism, when we're talking about cholera, the disease caused by Vibrio cholera, the classic description is rice water stools, copious amounts, many liters of fluids lost a day. In terms of the virulence factors that this species of Vibrio has, what are some of the toxins that contribute to this pathology? Yes, sir.
LT, that's the same as VTech, similar to VTech.
LT or what? CT, cholera toxin. But yeah, so in terms of mechanism, CT acts very much the same way as those E-type toxins. So you have dysregulation of the ions that flow in and out of those enterocytes and lack or loss of intestinal permeability, contributing to severe diarrhea. I will give you 50 of those. What you guys think?
C.
Good, okay. Okay, and again, the key word there is main route. Main route is still ingestion of contaminated water, food water, large numbers. And then also, sorry, just going back to probe this a little bit more, the symptomology is there, the rice water diarrhea. Don't usually encourage word association, but it is sort of really something you need to know. Rice water, as that description is, it's always associated or attached to Vibrio cholera. Severe dehydration is what you associate with the fatal outcomes. People basically become dehydrated and they go into shock. And then failing critical infrastructure is also associated in terms of epidemiology with Vibrio cholera and cholera outbreaks. Other Vibrio species, Parahemolyticus, we talked about a little bit, Molnificus and Altinolyticus. We do see these in the United States. What kinds of things are they associated with? Routes of entry, transmission. Uncooked shellfish, oysters, mussels, clams, things like that. What else? Wounds, I heard somebody say wounds. How does that happen? Yeah, so the acquisition occurs out in an environmental condition. So you're walking around wading in brackish waters, warm waters that the Vibrios like. You have an open cut, they come into that open cut, and can cause toxins. So ingestion, entry through open skin, ingestion of food and water are some common sources. And then the summer months is also sort of the high season for vibriosis. Parahemolyticus again, just normal gastroenteritis, fairly common, especially in those regional areas where you have Vibrio in those waters. Molnificus is really severe, so normal gastroenteritis if you ingest it, but also know that if you get it acquired through an open cut or wound, you can get this flesh-eating form of infections. It's really, really severe. Vibrio molnificus.
I have a question.
Yes, sir.
I noticed you went from Shigella to the Vibrio cholera. I was wondering if like Vibrio cholera, am I saying that right? Cholera?
Vibrio cholera.
Cholera, yeah.
Yeah.
Is that more significant than the other five that's kind of in between Vibrio, cholera, and Shigella.
So you're saying we skipped what?
We skipped like enteric Yersinia,
Yeah.
what was, I'm not good at saying these. Klebsiella, Enterobacter, Proteus.
Yeah, that's just for the sake of time, so we don't, show the 250 slides in block 3. But it doesn't mean that there won't be questions about Proteus, or couldn't be questions about Klebsiella. This review is not comprehensive, like none of them are comprehensive. So this is just sort of to zero you in on some things. So, but going back to your question about, well, you didn't really ask about this, but let's go there. With Providencia, Proteus, Klebsiella, some of those bacteria, what do we associate, what kind of routes of acquisition do we associate with those bacteria, and where do they colonize the human host?
Is that a question for the class?
I'm just looking at you, right, it's a question for the class.
Okay. Class.
Yeah, Megan. Say again. Yeah, you can definitely acquire the nosocomial, but in terms of where they might colonize somebody, yeah, they're primarily, so they're Enterobacteriaceae members, they're gut bacteria. And when we talk about opportunistic infections, particularly when people are already vulnerable, they're hospitalized or acquiring in the hospital, they cause a whole broad range of symptoms. So everything from UTIs, surgical site infections, pneumonia, broad range of things. When you're studying those, make sure that you know that those are Enterobacteriaceae, so gram-negative rods, opportunistic infections, gut bacteria. If there's anything distinctive about like how the bacteria are cultured, you might want to hit that. Proteus, for example, we hammered that pretty hard. You guys remember anything about culture? Yeah, it has that swarming appearance on media plates. And then in terms of motility, because of that swarming, what does that suggest about motility? It's like hyperflagellated, hundreds or thousands of flagella on those organisms. So Enterobacteriaceae, when we start transitioning over to Vibrio, Aeromonas, Campylobacter, maybe not so much, but Vibrios and Aeromonas, those are environmental bacteria. So you usually acquire them not so much in hospital settings, but when you're out in contact with water, where those bacteria are. Okay. And Aeromonas, this is not in the review deck, but what kinds of infections do we associate with Aeromonas hydrophila and other Aeromonas species? Okay, well just, yes, do go back and study that slide on Aeromonas, you would probably want to be familiar with that. Say again? Aeromonas, yeah, potentially. Yeah. Yeah, historically, association with leeches. Okay, Campylobacter, morphology of Campylobacter, so this is not a member of the Enterobacteriaceae, but you do find it in what kinds of sources of Campylobacter species? Say again? Poultry? Yeah, and lots of other food-producing animals. So this is a good chart to kind of be familiar with, but the common thread with all 4 of these species, virulent species of Campylobacter, you've got poultry, you've got pigs, cattle, sheep, dogs, cats, but with these 4, which is the most pathogenic, the one that causes most infections? Jejuni, yes, and that is mostly associated with which food-producing animal?
Chicken.
Exactly, chicken, poultry, very good. So with Campylobacter jejuni, we're talking about Campylobacter bacteriosis, which is a form of gastroenteritis, pretty run-of-the-mill, can be serious, can be severe, possible to get bloody diarrhea with this. Also, a sequela that you should be familiar with that is associated with Campylobacter jejuni infections Guillain-Barré syndrome, and that is true for Chichuni and Apsaliensis, those two. Morphology of Chichuni and these other Campylobacter species. Something kind of distinctive, right? It's not just a rod. It is a rod, but not a normal rod. Yeah, exactly. It's got like the gold-winged morphology. Number one cause of bacterial diarrhea in the United States, number one cause of gastroenteritis worldwide. Big deal. H. pylori. What kinds of problems does H. pylori cause? Stomach ulcers, gastric ulcers, gastric pain. Yeah, risk of cancer, and this kind of loop with GERD, gastroesophageal reflux disease. So esophageal cancer, stomach cancer, peptic ulcers. In terms of diagnostics, let's see. Histology, let's pick on that one. You want to biopsy some gastric tissue and get all this information on the top. What would be, it's not really clear from the slide, but what would be a disadvantage of just going in and grabbing some tissue from somebody's stomach lining?
Punching a hole in their gut.
It's a little invasive. Yeah, so there's some advantages to doing other methods if you can. What about just doing a stool culture? Somebody you suspect has a stomach ulcer? Yeah, probably not. Would not be expected to cover a lot of bacteria that way. Fecal antigen test, that is what's shown on the bottom left. That's just a normal immunoassay you can do. What would serology tell you if you wanted to just look at somebody's serum and see if they have antibodies to H. pylori? What would be some benefits and some limitations to doing that? Yeah. Exactly. So if they have antibodies, it's fairly common, right? A lot of people do carry this asymptomatically. So if they have antibodies to it, it's not necessarily going to tell you if you have an active infection, if they cleared it, or anything like that. But otherwise, serology can be useful if you just want to see somebody's been exposed to it. Alright, so now this is moving on to the fourth deck. Talked about Pseudomonas aeruginosa. This is another environmental bacteria, but remember that it can also cause, or it can also colonize even healthy people. And it is associated with a lot of hospital-acquired infections. So generally, the people who are most susceptible, there's another slide that I don't have in the review deck, but the people who are most susceptible to some of these infections are people who are hospitalized for things like burns and cystic fibrosis, who already have severe respiratory issues because huge problems for those folks. What's the gram morphology of Pseudomonas aeruginosa? On the fourth deck, so this is it's a gram-negative rod, very good. In the acronym ESCAPE, what does the P stand for? Great job, guys. Awesome. Oh, what else about Pseudomonas? Broad temperature range, and there's one other feature. It's not on this slide, but it is on this slide. What's another feature that really contributes heavily to its ability to cause some of these problems? Biofilm formation. Yeah, it is a big biofilm producer. And that contributes to its antibiotic resistance as well. Virulence factors. Okay, so you've got these exotoxins that are produced, can contribute to some of that tissue damage associated with people who have wound infections or burns, extra susceptible. Those unusual pigments that it produces And then just these run-of-the-mill invasive enzymes, elastases, proteases, lipases. Those all can contribute to some of these localized infections. Make sure you hit those skin infections, know that they cause ear infections, eye infections, but these are all opportunistic UTIs, opportunistic infections.
Question, ma'am.
Yeah.
Is that rash similar to the rose? Like, does it look similar?
It doesn't really, this is a type of folliculitis.
Okay.
The rose spots with typhoid fever are like non-blanching light, and if they're present at all, so yeah. That would have, and macropapular, whole body, that's exactly what that is. So this is the organism that's associated with people going in hot tubs, and because of the fact that it's so thermal tolerant and survives at higher temperatures, it can survive those higher temperatures and invade hair follicles, cause folliculitis. Big outbreaks in spas and things like that, especially the Salmon's original stuff. So what other things have we talked about that can cause folliculitis? Staph aureus, yeah. Okay. Burkholderia pseudomallei. What's the common source for this guy? Is this a normal commensal? Nope. Where does it live? Southeast Asia and Northern Australia, yeah. So there have been some rare incidents where we've isolated it in the United States, but it is associated strongly with the geography of Southeast Asia and Northern Australia. And then we talked about that bizarre case report where they basically had it aerosolized in this aromatherapy product, but that was a little bit unusual. Remember, if something is tagged as a biological drug agent, make sure you are able to associate it as a biological drug agent. So Burkholderia pseudomallei is one of those. It's capable of being weaponized. The disease it causes is what? Like what disease does Burkholderia pseudomallei cause? It starts with an M. Melioidosis. Yes, the great mimicker presents like a lot of different things depending on the route of entry. Difficult to diagnose, that's one of the reasons why it would make a good biological weapon. Acinetobacter baumannii. What does the A stand for in escape? Outstanding, very good. Gram-negative rod, right? But the morphology is a little bit different. It's Coccobacillus, stubby, almost cocci looking. Remarkable ability to survive in dry conditions, which makes it difficult to get out of hospital settings. Lots of intrinsic resistance. Just another run-of-the-mill opportunistic pathogen, except for the fact that it is so freaking hard to get rid of, associated with ESIs, UTIs, human surgical site infections, meningitis, septicemia. So kind of keep this in your head as opportunistic infections. And its nickname that it earned, even though it's been around for a long, long time, Iraqibacter, caused clusters of outbreaks in ships and ground units. H-duck, Haemophilus ducreyi. Anything interesting or remarkable about a gram stain from Haemophilus ducreyi? Stoolfish arrangement, railroad track arrangement. How does this present? and there's a special name for that ulcer called a chancroid, yes, very good. What other organism causes genital ulcers? Syphilis. Primary, secondary, tertiary? Primary. And then what is that called? Specifically, what's the term for a syphilitic ulcer? Chancre. And this is a chancroid. Has some differences. This has softer margins. Is this painless or painful? Painful, yes. And then what happens to the lymph nodes in that area, inguinal lymph nodes? Swell, become distended, and it's extremely, that's also painful. Common in the US? Where are you likely to see Haemophilus ducreyi chancroids? Asia and Africa, yes. Okay, I didn't put any Haemophilus influenzae slides there unless I put them later in the deck, but if I didn't, remind me to hit that with you guys. And actually, while we're at it, it's 8:52. Why don't we take a break and then we'll pick back up with what the product.
Yeah, WhatsApp or Signal, whatever you
It's a month.
That's the one you're at.
Like no clarity. The inside looks nice, but then look at the outside. Here's the gravy on the how do you have to stop here? This one of my first
like I'm doing
Sorry, these are a little bit out of order, but here, at least the Anthropomorphos slide is in their preview deck. So let's hit that real quick and then I'll backtrack. It might be a little bit out of order, but again, like just this is all in the fourth deck, kind of in the middle of the fourth deck, around slides 20 through 40. Alright, so H. flu. We have a couple different serotypes associated with it. There's one in particular that was very concerning. You guys remember which serotype? Type B, serious enough where we based our vaccine against H. flu off of it. So vaccination against HIB, the serotype B of H. flu is common. Be familiar with the very common non-invasive infections that you can get from both serotype B as well as other serotypes of Haemophilus influenzae as well. The upper respiratory tract infections and other infections of the like eyes and ears, common. Invasive infections, children are oftentimes impacted, and this is why we developed that vaccine against serotype B. To prevent meningitis, this is one of the big six. Hepatitis and laryngitis, pneumonia. I'm gonna skip back to where we left off with Bordetella. Again, I apologize if this is a little out of order. With Bordetella, it's fairly easy to study for because the symptomology is so unique, and you get that, unique presentation. Do we have a vaccine for this? Yes. A couple of you have asked me, like, as you're studying and, putting note cards together or spreadsheets or however you're choosing to do this, what kinds of information you should include. I do recommend if we have a vaccine against something like Bordetella pertussis that you take note of that. Sometimes, questions are which vaccine preventable disease, blah, 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 or something like a child presents with no vaccine history, you were to infer that they were not vaccinated against something like pertussis. Okay, gram stain of Bordetella pertussis. Gram negative rod, yeah. if you're going to treat pertussis, this is the difficult thing about treating pertussis, if you're going to treat, when would be the best stage to treat with antibiotics? Yeah, exactly, that first stage, because that's when the bacteria are actively replicating. In the proximal stage, remember, most of that pathology is due to the pertussis toxin that's expressed. So at that point, antibiotics aren't going to do much for the toxin that is causing the tissue damage. Members of Neisseria meningitidis, and Neisseria gonorrhea. These are gram-negative diplococci. Which of those species has a polysaccharide capsule? Meningitidis. Do we have a vaccine against Neisseria meningitidis? We do, definitely do. And it is against those different capsules that are associated with Neisseria meningitidis. So this slide here, we have a bunch of different capsule types. There's some epi association with the prevalence of serogroups A, B, C, W, Y, etc. So when we have our vaccine, which, oh darn it all, I did not include in this deck, but hit that slide that talks about the vaccine for Neisseria meningitidis, the tetravalent vaccine that is offered against Neisseria meningitidis, and that other one is that will cover type B. Diseases associated with Neisseria meningitidis, which two are we mostly concerned with? Meningitis is one of them, and the other one, meningococcemia, that distinctive one that is basically like a septicemia, bloodstream infection. That's why Neisseria meningitidis. For the epi, I think the reason why this is a good slide to hit is to make sure you have an understanding of transmission and populations where this is spread. So Neisseria meningitidis, we are talking about a lot of transmission with young, healthy people, people who are in barracks. Not that we are unvaccinated, we're vaccinated against it, but close contact, younger people, daycares, schools, things like that. The BSDI, if you have somebody who you suspect has gonorrhea, what would be their presentation?
Virulent discharge.
Virulent discharge, yep. Anything else? What's dysuria?
Painful urination.
Okay, very good. Yeah, so painful urination, dysuria, urethral discharge. Specimen type, if you want to do a gram stain, you can use the exudate in that urethral discharge. And then what would you see on a gram stain of an exudate from a patient who has Neisseria gonorrhea? Gram-negative diplococci. Yep. This slide, make sure you study this. A couple questions that you could craft from this slide would be, it's sort of like a summary slide, so you don't have to hunt through multiple decks to find it, but if you have a, 35-year-old patient who has meningitis, two different types of bacteria that would rise to the top of your differential, what two types of bacteria would you suspect? Strep pneumo and Neisseria meningitidis, yeah. If you have somebody who's very old, what additional bacteria would you suspect? Listeria, yeah. And then for the younger kids, that's when it gets kind of crowded and you have a bunch of different things. Very young NMEC, neonatal meningitis, E. coli, Listeria again, GBS, group B strep, and then H flu serotype B. Up at the top there, if you have CSF from any patient of any age, as meningitis signs and symptoms, take CSF. Any organism that you find in there is going to be relevant. So any question that asks you to look to know what the morphology of those big six are, is gonna be very relevant, not contrived, relevant stuff. So if you find gram-positive cocci, what are some things you'd be thinking of, and what are some things you'd rule out for etiologies? Do you think it would be Neisseria meningitidis if you had gram-positive cocci? No. Do you think it would be H. flu? No. What could it be? Herpes strep or strep pneumo. And if it's like a 15-year-old and not a 2-month-old, then you're leaning more towards strep pneumo. Okay, so make sure you're studying that and putting those pieces together to be able to answer questions about that. Okay, Bartonella, Kinsella, cat scratch disease. Source of transmission oftentimes? Cats, what specifically about cats? Scratch, bite, yeah. If they actually bite you, and then one other, somebody said fleas. Yeah. Okay. In most healthy people, that presentation is gonna be more just regional lymphadenopathy. The initial entry site might have a skin papule develop. Bacillary angiomatosis, this is more associated with what kind of population? Immunocompromised, yes. Before I move on to Brucella, with that genus Bartonella, Brucella was only one of 3 that we talked about. What were the other 2 species we talked about? Quintana and Bacilliformis. What is Quintana also known as? Trench fever, and how is that transmitted primarily? What about the lice? How is that transmitted from lice to humans? Feces, yeah. Feces that you then rub in that have those bacteria in them. But with Bacilliformis, how is that transmitted primarily to humans? Sandflies, and that causes what kind of presentation or presentations? Okay, so yeah, exactly. The acute phase and then the chronic phase. The acute phase is known as what? Peroneal fever. And then the chronic phase? Peruvian warts. Yeah, which is the one that has the high mortality rates? Peroneal fever. Yeah, and the whole package is carrying on disease. Are sandflies the only way to get carry-on disease?
Mother and child.
So what did Daniel Carrion show us? Yeah, you can inoculate yourself with a pus from one of those Peruvian warts and contract Peroneal fever as well. So sandfly transmission is a primary mode of transmission. You can also inoculate yourself as well with the bacteria on it. Brucella. Morphology here, biological threat agent, gram-negative Colobacillus, slow growing, all good things to know. Maintained in what kind of environment? Where would you find Brucella? What do you associate it with? Livestock, yeah. Yep. Let's see if there's anything else that's tricky here. Tularemia, Francisella tularensis, another slow-growing gram-negative rod biological threat agent. Make sure you know this one. Some routes of transmission for Francisella tularensis.
Rabbits and ticks.
Rabbits and ticks, yes. And can also be weaponized, aerosolized. Forms of the disease here, glandular respiratory form, nodular lymph node involvement, typhoidal, where you have this entire systemic effect, and pneumonic tularemia. Depends on the route of entry. Can you get this in the United States?
Yes.
What regions in the United States would you associate this with? Kind of heaviest cases are in those tick infested areas, forested areas. Similarly, Yersinia pestis, can you get this in the United States? Okay, and then what part of the United States do you associate with plague? Western, yeah. We talked quite a bit about the presentations of plague and routes of transmission. If you get bubonic plague, how are you usually getting bubonic plague? Flea bites, so yeah, primarily flea bites. How do you get pneumonic plague? So the source of those aerosols, let's say it's not a biological weapon, it's just a natural source, where would you get pneumonic plague? From somebody who has pneumonic plague, right, they're breathing on you. How did they get pneumonic plague if they didn't have a biological weapon or they weren't in contact with somebody who had pulmonary or pneumonic plague? They probably had bubonic plague and then it progressed to pneumonic plague. So you can get progression from bubonic plague to pneumonic plague. Sorry, I'm just this notes real quick. With the name bubonic plague, what does the word bubonic indicate? Does it mean bubos? And what are bubos? They are lymph nodes that are distended and swollen. Bubos, painful, distended, swollen lymph nodes. Knowing that this is a gram-negative Coccobacillus, you can culture it, is important. With these arthropod-borne bacteria, they usually have the ability to grow at cooler temperatures, otherwise they wouldn't be able to survive in that arthropod host, which has a cooler body temperature. In this genus of Yersinia, in depth 3, there were a couple of other organisms that were also in this genus. You guys remember which organisms are also Yersinia? One species name starts with E and the other one starts with P. Pseudotuberculosis and Anacolytica. And those cause what diseases generally? Yeah. Yep, and gastroenteritis. Yeah. Syphilis. The hardest thing I think with syphilis is the lab diagnostics, but there's also a slide that you want to make sure you study about the 3 stages of syphilis in the presentation, those 3 stages. For diagnosis, if you had somebody, particularly if they had like an initial primary syphilitic symptom like a chancre, genital ulcer chancre, come in. For diagnostics, you have an algorithm where you don't immediately proceed to doing any, like definitive test, but this algorithm entails a non-specific screening test, and then you have to follow that up with a specific confirmatory test. So on this slide, these are the direct methods, microscopy, you can do direct methods, microscopy, looking for the organism. You can also do these serological based tests where the screening test would be this first one, the non-treponema test, where it is super sensitive, right? That's a good screening test. I wanna make sure you guys get it. Super sensitive but not specific. So a whole bunch of false positives are possible with this, because what you're looking for is a reagent. You're looking for evidence of damage that would be caused by the treponemes in this infection. Okay. If you get a positive result on a non-treponemal screening test, this indirect test, then you have to do a confirmatory direct treponemal test to make sure that you have a specific diagnosis, that you actually have Treponema pallidum. And that test would look for the organism itself. Okay, Borrelia species. So we have a bunch of different species that can cause relapsing fever. Make sure you understand that you have different vectors associated with epidemic or endemic forms of relapsing fever. And that influences the geography and the endemicity of these diseases. And also the presentation itself, we talked about this, what relapsing fever means and what's going on when you have relapsing fever, when you have fevers that are spiking and then you have clearing of the organism and then spike again and clearing of the organism. That is caused by what? Antigenic radiation, yep, exactly. Okay, the last spirochete that we talked about is Leptospira interrogans. Where would you expect to find this organism, cases caused by this organism? In the tropics, usually, yes, but you do occasionally get some, unusual cases in other parts of the country as well, like that one I mentioned in Illinois in 1994, talk about it does happen, associated with things like adventure sports, exposure to water. What is in the water that exposes you to this bacteria? Urine, usually from animals who are carrying Leptospira interrogans. In terms of presentation, the more severe presentation, what kind of symptoms would you associate with the more severe form of leptospirosis? Jaundice. Also, what's the common name for the severe presentation of leptospirosis? Weil's disease. Very good. You said jaundice. What else would you look for? Bloodshot eyes, cough, diarrhea, vomiting, that rash over the lower extremities. Okay, moving on to deck 5. We've got anthrax. We talked about several different forms of anthrax. What are those 3 forms of anthrax?
Pulmonary, gastrointestinal diseases.
Great, okay. Good job. Pulmonary anthrax, hallmark of pulmonary anthrax? And diagnostically, what would you use to show the widened mediastinum? Chest X-ray. What kind of morphology are we talking about? Let me back up a little bit with Bacillus anthracis. If you were to see gram samples. Gram positive, did you say rods or conci? Rods, great. Anything else remarkable or distinctive or unique about the rods? Spore formers, very important. Spore forming bacteria. Bacillus cereus. So same genus, also gram-positive rods, spore forming. 2 different forms when you're talking about ingestion. Make sure it's very similar to Staph aureus when we're talking about the toxin-borne or the toxin-mediated form. And B cereus with this emetic form, which is primarily toxin-mediated, and because of that, you have a short incubation period because there's really nothing to incubate. You have a preformed toxin that's in the food, and that food is usually what? Fried rice. That's like the classic food that's implicated with Bacillus cereus. Or a true infection where the bacteria are not destroyed because you have definitely not heated the food outside of that danger zone, so the bacteria are present, and you ingest those bacteria and you get what kind of symptoms with that form. Okay, so then you would actually have a legit incubation period, and it would probably take much longer for symptoms to start appearing. Clostridium difficile, you want to have a pretty good grasp of what's on this slide. Make sure you know about this association with antibiotics, particularly broad spectrum antibiotics, and the dysbiosis that can be caused by broad spectrum antibiotics. This is happening in the gut, clearing out beneficial bacteria and allowing the overgrowth of C. diff. What's the hallmark of severe C. diff infections? Pseudomembrane, yeah. Forms in the colon. Clostridium perfringens, what are some things that can be caused by this organism? Some diseases, infections.
Oh, yeah, yes, food poisoning.
Gas ingredient, food poisoning, yep. And there's another one that's really
Big bug.
necrotizing enteritis, yes, necrolidins. Yep. Practice question for you guys, you haven't had a question in a while, wait for a little bit. What do you think? Okay, I think E, because we just studied that slide, which is great, but let's back up a little bit and figure out why we would be ruling out some of these other etiologies. So we talked about Vibrio vulnificus and the fact that it could cause some flesh-eating disease as well, severe tissue damage. What in this question stem would allow you to eliminate that as an option? It's not spore forming. Is it gram positive? No, it's gram negative. In fact, for Vibrios, what's the morphology like? It's a bit curved, right? So curved gram negatives, definitely not spore formers. The fact that this question tells you it's a spore former actually allows you to eliminate everything else except what other option? Clostridium botulinum. Yeah, so if you know that, then you can eliminate all but two, and then the gram positive, you need to be able to know that too. So just because a lot of these might be able to also cause food poisoning, like Staph aureus can certainly cause food poisoning, we know that, we've just studied that. It can also definitely cause soft tissue infections. So if you don't know both of those pieces of information, you get a question like this wrong. Okay. Those other presentations we talked about, enteritis, necrolidins, the back alley abortion, clostridial metritis infections, food poisoning. Yeah, so make sure you kind of hit this slide as well. Clostridium botulinum, causing botulism. There are a bunch of different forms of botulism. I have a pretty good sense. I think you guys will do fine on this. With most of these, what is the route of infection?
Canned food?
Well, yeah, I'm not phrasing it the way I want to. Like if you have a wound, then it would be coming in through broken skin, right? Bacteria. What is actually coming into the skin usually? Not the toxin, the spore. Yeah, so sporulated bacteria, and then when it gets inside of you, then it will germinate oftentimes with most of these presentations. So, yeah, it's usually the spores. Let's see, what else on this slide? Iatrogenic, you guys comfortable with that word and what it means?
It's like provide.
Caused by seeking medical care or medical attention, even if it's cosmetic. So it's not like a surgical site infection, it's like a completely accidental acquisition of a disease or some pathology. Iatrogenic botulism is just like it says in the explanation. So you go in for some purpose and you get this form of botulism. What is the toxin that is associated with these different presentations of botulism? Yeah, not a trick question, botulinum toxin. Very good, Botox, Botox. And it is a neurotoxin. Tetanus, also, usually students do really well on this question. Common in the United States. We have a vaccine against it. If we didn't, it probably would be common, but we don't get that many cases because it's covered in Tdap or DDaP vaccine. Yeah, the symptoms, I don't even think we need to go over this, they're just classic symptoms of tetanus, the lockjaw, the muscle spasms. Usually diagnosed just based off that presentation. Yeah, I think I covered everything I need to cover on that. Listeria monocytogenes. This one is loaded. 3 populations, 3 different presentations. Healthy people, what kinds of symptoms are associated? Non-invasive gastroenteritis, so you could lump that in with many other organisms that we've talked about in healthy people, but main point, self-limiting diarrheal symptoms, usually gastroenteritis. What populations are we more concerned about? Yep. And with pregnant women, when we're talking about the ability of this organism to cross the placental barrier, cause infections or problems in the fetus, what kinds of problems are we most concerned about? Meningitis, miscarriage, premature delivery. Yeah. Do we have a vaccine against Listeria monocytogenes? Nope. So, we put a lot of emphasis on what in order to keep cases of meningitis and other invasives. Avoiding these dairy products that are implicated oftentimes, yeah. This is Listeria monocytogenes. And deli meats as well. The fact that it grows at colder temperatures is important too for those populations. Is this a spore former? It is not. The only 2 genera that are spore formers are what? Okay, good. Okay, getting near the end, guys, almost done. Cranibacterium diphtheria. Do we have a vaccine for this? Yes. Is it common in the United States? No. It is not common in the United States. We did have a case last year, but before that it was like 20 years. Yeah, we practically eliminated it. When people do have cases of diphtheria though, what's considered the hallmark symptom of diphtheria? Yeah, a pseudomembrane that can form in near the back of the throat, uvula, tonsils, things like that. What can actually kill you with diphtheria? Airway obstruction.
Slough off, especially with children.
Yep, exactly. So that pseudomembrane, it sloughs off, it can lodge in the airways. And then also that cutaneous form of diphtheria as well. Still has that grayish, unique pseudomembrane, even with cutaneous diphtheria. Right, last two slides, this one is Cutibacterium acnes, last one is Lactobacillus species. Cutibacterium acnes, just kind of throwing it in there as yet another example of a bacteria that has some of these invasive enzymes, things like lipases that can contribute to inflammatory response in that local sebaceous gland hair follicles. Gram-positive rod, normal flora. And then lactobacillus. Why does lactobacillus play such an important role for human health? Probiotic is important for maintaining normal proper balance of bacteria in the gut, in the vaginal tract. So it's a good bacterium. And then we talked about one other good bacterium, like really, really good bacterium. Bifidobacterium, yeah. And what was the morphology on bifidobacterium? Yeah, it's bifurcated, yep, and that's what the name bifido means, bifurcated. What about the gram stain? Gram-positive rod, bifurcated. Okay, that's it. Those are the main things. I think you should be in good shape. Parting notes for you guys, parting thoughts, and then I'll open it up for questions. So Dakota, you had asked about full spellings of things. I will, make sure that the full organism name with no cute nicknames are out there, so the entire organism. Something really lengthy. Basically what I was just stressing earlier, especially with sterile specimens, you really do have to rely on those morphologies, and again, that is not confined. That is real world implications. I know it's a lot to study, but the way that's arranged in those decks 1, 2, 3, and 4, 5, should help you kind of study them according to those gram stains, so you can get that in, and then just kind of continue to, this is the way I study material, but I start pretty broad, and then I get more and more specific until I get pretty comfortable with the organism itself. Like I said, make sure if it is something we vaccinate against, make sure you're tracking that. If we have some way or major strategy of mitigating those infections, make sure you're kind of honed into that. Presentation is the name of the game, but not all presentations are significant. If it's just a normal diarrheal bug or causes gastroenteritis, it's kind of like, we can't really make it something special. A lot of different organisms can cause diarrhea, a lot of different organisms can cause UTIs and wound infections. So kind of look for something distinctive. Do you guys feel pretty comfortable about being able, now that we've gone through and done that review, pulling out some distinguishing factors and characteristics of these different bacteria, and how you would need to do that on an exam, and how you'd have to rule out options based on how the question is built? Okay, seeing some head nods. What questions do you guys have? Yeah.
I have one, ma'am. It's pretty minor, but on the slides, the Staph aureus slide says that impetigo is more likely caused by Strep pyogenes, but the Strep pyogenes slide says impetigo is more likely to be Staph aureus.
Yeah, I'm sorry, someone else had pointed that out too during the break, and I will have to look up which one is accurate. I think it's Staph aureus is more common, and then Strep pyogenes is the classic form of impetigo. But it doesn't matter for the exam, even though I shouldn't have mistakes in there like that. I appreciate you pointing that out. What does matter for the exam, specifically with impetigo, since it can be caused by both Staph aureus and Strep pyogenes, is knowing that the presentation is not clinically distinctive between those two. So if you want to diagnose it and get an ID, you have to take a sample of that crust and submit it to lab and have them grow it up. That's what's important. But both of those organisms are significant causes of impetigo. Okay, so good question. Any other questions? Okay, I'll be down in my office. Let me know if you want to come and see me over lunch today. I think tomorrow is gonna be a little hectic. I'll be on site for the clinical inquiries for the seniors, but it will be kind of a packed day. So if you do wanna go over material, today would probably be a better day for that.
Would be around Thursday, ma'am.
Yeah, I'll be around Thursday as well. Yep. Okay, guys, well, thanks for your attention. I hope that helped a little bit. I hope over the 4th of July, you guys thought of Campy when you had chicken. I was definitely thinking of chicken. My family made a bunch of barbecue stuff. I was just staring at it with some level of distress. Deviled eggs and I think all sorts of things. So I was like, oh no. So, alright guys, I will, if I don't see you before then, I will see you Friday at 8 o'clock. Thank you.